Specification number 13 tells us to outline the role of bile in digestion. Bile helps by emulsifying fats, that means it breaks large fat droplets into much smaller ones. This increases the surface area, making it easier for the enzyme lipase to chemically digest the fats into fatty acids and glycerol. From there, the small intestine absorbs these products of digestion. The nutrient molecules then travel through the hepatic portal vein straight to the liver, where the liver carefully monitors and adjusts the blood content. Finally, the blood leaves the liver through the hepatic vein and enters the general circulation, ready to be transported around the body. Now let's talk about the role of bile. Bile is produced by liver cells and then stored in the gallbladder until it's needed. It has two main roles. First, neutralization. Bile is alkaline, so it helps to neutralize the hydrochloric acid coming from the stomach. This is important because the enzymes in the small intestine work best in an alkaline environment. Second, emulsification. Bile breaks large fat droplets into much smaller ones. By doing this, it increases the surface area, which allows the enzyme lipase to work more efficiently, breaking fats down into glycerol and fatty acids. When we talk about the emulsification of fats and oils, it's a bit like shredding a large sheet of paper into lots of smaller pieces. The paper is still paper, it hasn't changed chemically, but now it has a much larger surface area. In the same way, emulsification is a form of mechanical digestion, not chemical digestion. It breaks large fat globules into much smaller droplets without altering the chemical bonds or structure of the fat molecules. Chemical digestion, on the other hand, is different. It actually changes the chemical structure of molecules. For example, lipase breaks fats down into fatty acids and glycerol. Here's how bile helps with this process. Each bile salt has a hydrophobic side that attaches to the fat globule, and a hydrophilic side that interacts with water. This action splits the large fat globule into many smaller emulsified droplets, giving enzymes like lipase a much bigger surface area to work on. Imagine starting with one big fat globule. On its own, it doesn't give much surface area for enzymes to work on. Then bile salts come in. Each bile salt has two sides. The hydrophobic side sticks to the fat, while the hydrophilic side faces the watery surroundings of the intestine. As the bile salts attach, they pull that big fat globule apart into many smaller droplets. These are called emulsion droplets. The fat itself hasn't changed chemically, it's just been broken into smaller pieces. Now, because there are many more small droplets, the total surface area is much bigger. This gives lipase enzymes plenty of space to act. Lipase then breaks the fat into fatty acids and monoglycerides, which group together into tiny micelles. These micelles are small enough to be absorbed through the walls of the small intestine and passed into the bloodstream.